আচ্ছা, তো আইসোলেশন এন্ড ক্যারেক্টারাইজেশন অফ ফাঙ্গি এর মধ্যে যেটা আছে সেটা হচ্ছে যে ফাঙ্গি কি জিনিস? ফাঙ্গি হচ্ছে একটা ইউক্যারিওটিক এবং তার সাথে ফিলামেন্টাস ব্রাঞ্চড স্পোর প্রডিউসিং একটা মাইক্রো অর্গানিজম। এখানে ল্যাক অফ ক্লোরোফিল আছে। দেন সেল ওয়ালস হচ্ছে কাইটিন সেলুলোজ এবং এর সাথে বোথ এখানে থাকতে পারে। যদি আমি ফাঙ্গাসের কথা দেখি তাহলে ফাঙ্গাসটা কি জিনিস? ফাঙ্গাস হচ্ছে একটা ইউক্যারিওটিক। ইউক্যারিওটিক হচ্ছে ফাঙ্গাস। আর এটা হচ্ছে ফিলামেন্টাস থাকবে ব্রাঞ্চ অবস্থায় এবং স্পোর প্রডিউস করার মতো একটা মাইক্রো অর্গানিজম। আর ল্যাক অফ ক্লোরোফিল এখানে আমি দেখতে পাবো। সেল ওয়ালস গুলো কাইটিন সেলুলোজ এবং বোথ এখানে থাকতে পারে। আচ্ছা, তো তারপরে যেটা আছে সেটা হচ্ছে যে অল ফিলামেন্ট অল ফিলামেন্টস আর কল্ড দ্য হাইফি। ফিলামেন্ট যেগুলো আছে সেগুলোকে বলা হয়ে থাকে হাইফি বলা হয়ে থাকে। এক বছরের যেটা আছে সেটাকে হাইফা বলে। হাইফা আর এর হচ্ছে প্লুরাল ফর্ম হচ্ছে হাইফি। Each hypha consists of one or more cells surrounded by a uh, tuber cell wall. Tuber cell wall. So a mass of hyphae make up the body of a fungus, which is called a mycelium. Plural mycelia. The hyphae of the most fungi are divided into cells by internal walls called septa, singular septum. Then hyphae. Hyphae, this is septa hyphae, septed hyphae. Then in coinocytic, সিনোসাইটিক যে হাইফি সেটা। তো তারপরে যেটা আছে সেটা হচ্ছে কি হোস্টোরিয়া। এখানে হোস্টোরিয়ার একটা ফিগার এখান থেকে দেখা যাচ্ছে। তো তারপরে হচ্ছে কি যে রিপ্রোডিউস বাই স্পোর্স। স্পোর্স আর রিপ্রোডাক্টিভ সেল। এটা যেটা আছে সেটা হচ্ছে সেক্সুয়াল, সেটা হচ্ছে মিওটিক ইন অরিজিন অ্যান্ড অ্যাসেক্সুয়াল ইন মায়োটাইড ইন অরিজিন। তো সেক্সুয়াল যখন হবে সেটা হচ্ছে কি মিওটিক ইন অরিজিন অ্যান্ড অ্যাসেক্সুয়াল হচ্ছে মায়োটিক ইন অরিজিন। দেন ফর্মদের মধ্যে ফর্ম হচ্ছে ডিরেক্টলি অন হাইফি অ্যান্ড ইনসাইড এসপোরেনজিয়া অ্যান্ড ফ্রুটিং বডিতে এগুলো আসলে ফর্ম করে থাকে। না, তার হচ্ছে কি আছে অ্যামেনিটিয়া অ্যামেনিটা ফ্রুটিং বডি। দেন হচ্ছে কি যে পিলোবোয়াস স্পোরেনজিয়া and penicillium hyphae with conodia. দেন মাশরুম লাইফ সাইকেল যেখানে আছে সেখানে হচ্ছে যে এখানে নিউক্লিয়ার ফিশন ইন ব্যাসিডিয়াম। দেন ইয়াং ব্যাসিডিয়া দ্য অনলি diploid cells, meiosis. Then haploid mycelium, then hyphal fission of haploid mycelia, then mycelium and fruiting body, and the dikaryotic um, thing is present here. Then it is a sign of haploid, and this is the diploid n plus n. Diploid is 2n. Then human fungus interactions. Human fungus interactions, beneficial effects of fungi, decomposition, nutrient and carbon cycling. Then biosynthetic factories can be used to produce the fungus, produce the drugs, antibiotics, alcohol, acids, food, fermented products and mushrooms, model organisms for biochemical and genetic studies, model organisms for biochemical and genetic studies. Then harmful effects of fungi, the destructions of food, lumber, paper and cloth, and animal and human diseases including allergies and toxins produced by poisonous mushroom within food, grains, cheese, etc., and plant diseases. It is the harmful effect of fungi. Then isolation and identification of fungi. Fungi can be isolated from virtually any place on Earth, from tropics to the poles, and from the mountain tops to deep oceans. Fungi can be isolated, and in our laboratory we'll focus on airborne fungi, soil fungi, infected fungi, then water fungi. First of all, airborne fungi. As we know, fungi are present in air in spore shape, but sometimes we find hyphae in air. The main function of the spores is conserve of species and distribution. The functions of spores is conserve of species and distribution. Then the function, then the factors affecting on the distribution of the fungi: number one, temperature, speed of the air, rains, humidity, cycle of spores production. This can be happen. Then method of exposing the petri dish: certain fungi are more likely to get their spores into air than others, and airborne fungal spores can serve as the infective agents of land disease and may also be allergenic. The simplest technique for airborne fungi involves the horizontal or the vertical placement of the petri dish containing the nutrient agar, so that they trap any spores that fall on or bow into them. Usually 30 minutes to 3 hours out of doors is required for a good sample using this technique. Usually 30 minutes to 3 hours out of doors is required for a good sample using this technique. Then isolations of the microorganism from the air. Yeah, pour the melted agar medium on the sterilized petri dish. Then we can get this. Then allow the medium for solidification. Remove the lid and expose to the air for 5 to 10 minutes. এটা 5 to 10 minutes এর জন্য কি করতে হবে? এক্সপোজ করে দিতে হবে। দেন তারপরে কি আছে? ইনকিউবেট দ্য পেট্রি ডিশ। এই পেট্রি ডিশ যেটা আছে সেটাকে ইনকিউবেট করে দিয়ে তারপরে এখান থেকে অবজার্ভ দ্য ব্যাকটেরিয়াল কলোনিজ ওভার দ্য অগার মিডিয়াম। এবার অগার মিডিয়ামে এদের যে কলোনি যেগুলো আছে সেগুলোকে অবজার্ভ করতে হবে। দেন পারফর্ম ডিফারেন্ট বায়োকেমিক্যাল টেস্ট ফর identification of the bacterial colonies. Then we can get this kind of thing. Then soil fungi. There are many techniques for studying the soil fungi, and the choice of methods are based on the aim of studying. To collect the sample, you must take 10 to 15 centimeter of the soil approximately, and 50 to 100 gram by clean towel. Then store the sample in the clean bag. To collect the sample, you must take 10 to 15 centimeter of the soil approximately, and 50 to 100 gram by clean towel. Then store the sample in clean bag. In clean bag, studying of soil fungi, direct method and indirect method. Studying of soil fungi, direct method and indirect method. Then direct method: as a result of non-transparent soil component and randomly distribution of the spores, it's impossible to study the soil directly under the light microscope. So there are samples. There are sample methods for this. So as a result of non-transparent soil component and randomly distributions of the spores, it's impossible to study the soil directly under the light microscope. So there are samples method for this aims as using of contact slide method. For studying the natural distributions of the soil, take clean glass slides and put them in different sites in soil for 1 week. After that, bring the slides, dry, fix them, and flame them, wash the slide, and finally staining these slides for microscopic examination. And this method using for studying the distributions of the hyphae in soils, but it is not useful for identification and the types of fungi due to not available of spores. Then soil smear preparation, smear method: by using the clean glass slides and press it in soil, then stain these slides with the lactophenol cotton glue. Then indirect method, dilution plate method. This is the dilution plate method. Then soil plate method, strewing 0.05 gram of the soil on the nutrient media, complementary with antibiotics. Then this is the method we can see here. Different method. Then isolation from the infected plants. Here is 60 second, 5 minutes, 2 minutes, 2 minutes. So 3.5% sodium perchlorate here we can see. Then water. Then dry in the clean bins, and then plate pieces on the PDA and MEA. Then fungi were isolated from collected. Fungi are isolated from the collected root samples, and first samples were washed with the tap water to remove the soil debris, and then immersed in 1% sodium hypochlorite NaOCl for 1 to 2 minutes, and samples were transferred to settle the distilled water SDW for 2 to 3 minutes, followed by drying on the sterile filter paper. The root were cut to small 2 to 3 millimeter pieces, and 4 pieces were placed on 2.5% potato dextrose agar by PDA medium amended with 10 milligram uh, 10 milligram per liter rifampicin and 200 milligram per liter of ampicillin. Ampicillin, the plates were kept on the room temperature in laboratory for 7 to 10 days, and fungal growth on each plate or subculture to a new plate, and the plates were kept in the incubator at uh, 27 to 29 degrees Celsius, and the growth of the fungi were observed daily. And this is the figure. You can see total process of this figure. And then subculturing make a subculture from the old colonies of the fungi as follows, and obtain a obtain a metal loop on needle and flame it until it turns red hot. Wait for 10 seconds for loop on needle to cool down, then using the loop on needle according to which you will remove an isolated colony of fungi. Aspectically, transfer the cell colony into the nutrient media, incubate the plate at the room temperature for next week. Isolations of the fungi from the contaminated air. We can see here the CL dilution. We can see, then we can see the result, isolation from the pathogenic fungi from the plant parts, then PDA plates, then microscopic structures of the isolated fungi and spores appears for different things. Then methods of identifications of fungi. Number one, weight mount or tease mount method for fungal hyphae identification. Procedure of the weight mount preparation: take a grease free slide and plate with fungus culture with the help of a sterile scalpel or 90-degree bend wire. Remove the fungal colonies from the plate, which might contain a small amount of supporting agar. Place the portion of the culture into a slide to which has been added to a drop of lactophenol cotton blue or aniline blue. Place the cover slip into the position and apply the gentle pressure to dispose the general growth and the agar and extreme microscopically. Drawbacks: characteristics: arrangements of the spores might be disrupted when the pressure is applied to the cover slip. Then adhesive scotch tape preparation for fungal spore identification. Then procedure for scotch tape preparation: touch the adhesive side of a um, uh, cellophane tape to the surface of the colony. Adhere to the tape to the surface of a microscopic slide to which has been added a drop of the lactophenol cotton blue or anile blue. For the characteristic shape and arrangement of the spores, microscopic examination is required, and this method allows to observe in a way it sporulated in culture, and easy method to identify the organisms. In instances where the spores are not observed, a wet mount should be made as backup step. A wet mount should be made as a backup stage. And then the microslide culture technique for fungi identification. Procedure of the microslide culture technique: take a sterile petri dish and remove this lid, then place a sterile filter paper over it and add the distilled water to moisten the filter paper. Then put the glass slide over the glass rod and add 5 millimeter square PDA agar on glass slide from agar plate and incubate the spores of fungi on the agar and cover with the cover slip, and incubate at the room temperature for 7 days and remove the cover slip and mounted the next slide and observe under microscope. Then cover slip culture technique for fungi identification. This technique is simply less time consuming technique which produce high quality permanent mounts and is suitable for clinical isolation, identification, student teaching, and examination of fungi at different stages of their development without disturbing the arrangement of the spores and hyphal structure. It is advantageous over the slide culture technique that if the first preparation fails to demonstrate adequate sporulation, there are still left to be examined weekly in intervals, and the ability of the aerial mycella to adhere to a glass surface has been utilized as a basis of this technique. And then procedure of the cover slip culture method: insert 4 to 5 cover slip on the petri dish containing POA at an angle of 45 degree, and inoculate organisms at the point of media and cover slip. Incubate the plate at room temperature for 7 days. After incubation, cover the cover slip, remove the cover slip gently and mount it with the lactophenol cotton glue and observed under microscope. So these are the references we have taken this thing.